So we'll just start off by telling you kind of where we started in our collaboration and relationship and where things are going running the central manufacturing facility. So as Kirsten mentioned, we started out with vans, and so the goal of vans was to manufacture a COVID vaccine for New Zealand if required, whatever that looked like. Maybe that was, you know, someone like Pfizer BioNTech coming to New Zealand and wanting to make their things here, or developing our own. So is this laser going to work? So we basically, not point you in the eyes, started with trying to optimise both the mRNA and the lipid nanoparticle side of things. So we started out looking at the antigen, converting the mRNA to the protein, so comparing mRNA to the same protein vaccine, converting to mRNA. We would look at the immunogenicity and expression, and then optimise for cross-protection of various strains was kind of where we ended up with VANs. And alongside particle development. So we first looked at the commercial LNP properties, and then we optimised those, and then we started developing novel lipids for the LNPs. And so to do that, we developed this process where we would order a gene and then go through this sort of cycle where we start to generate the mRNA. So starting with the DNA template through to RNA production and analysis. So we had to develop all these techniques, encapsulation of the mRNA, the in vivo evaluation, and then optimization, and always with the idea that we would transfer to South Pacific Sierra when it was time to do that. So just to give a bit of Becca and I's CV, so we have made lots of constructs together, so we've made over 70 things, so 70 vaccines. More than 200 vaccines have been tested, so that was 70 different things and 200 vaccines. And we've done a bit of outreach, so we've given a few talks and published a few articles and posters. Over to you.
Sorry about the font changing, but you'll get the main points. So just to talk a bit about the team structure of the central facility, we have kind of made sure that we have a routine team, we like to call them, so people who will make sure that you get what you need from us when you need it if you're running a project with us. We have a development team which are more involved in the troubleshooting when things go wrong, so people with a bit more experience and molecular skills, for example, design of new constructs when they come in, and process development, because we want to always be able to change to what people in the RNA world are changing to and bring in new technologies and make sure we have the space to do that. And we have a wonderful immunogenicity team, because every time we make changes to our process, we also need to check that everything is still giving the response we expect, and that we can make these changes confidently before we send things out to you. So just to put some actual faces to those bullet points, these are the wonderful people that we work with, we're very lucky to have, and a lot of people have been involved all the way from the VANS project that Sarah mentioned until now. And so as a production facility, since we started officially in sort of April this year, we've produced 88 RNA platform products, which we're quite proud of. And this includes mRNA. Some people just want naked mRNA to transfect cells with, mRNA that's encapsulated, and oligonucleotides as well, and those can be encapsulated too. So we're making some progress. And these are the fast starts that were mentioned by Kirsten and Gary. So a lot of these people will talk today, so I won't go into detail, but it's just to show we're about halfway through the program now. So the fast starts were 1 year grants, which started sort of staggered due to contracting and things that take longer than you think. And some, like the novel lipids, you can see we've made a lot of progress. So that's making RNA, encapsulating and testing those. And some are at 5% because of roadblocks, such as getting staff, or, for example, with RNA building blocks, which is a project to make NTPs ourselves in New Zealand. They've just been made, and now we come into action where we are going to make RNA with those and test out whether they work the same way. So that's just an overview of how we're going. So I think we're also about halfway through those.
So with our vans work, and then especially the last year of the platform with having to adapt to all these fast start projects, we've increased our capabilities quite a lot, and we can do different things. So we can do a range of RNA payloads, including mRNA and oligonucleotides. So and some of those oligos do include DNA. We have now got capability to make lots of different nanoparticles for different things. So that might be because you want to have long-term storage or incorporate some of the novel lipids that Gavin will talk about later. And we can sort of change the recipe depending on what you need. We can incorporate small molecule additives, especially into the LNPs, but we can also do that with oligonucleotides and soon mRNA, so dyes and things like adjuvants. We have a bit of experience with protein design, so we can help you, if you've got a target, we can help you design your protein and then therefore your RNA. And we can also do expression studies, so make sure that the construct actually works. And so we have a development team, and so that's obviously to try and advance the progress of our team. And so some of the projects that we've been working on is throw my remote away. So some of them are already in progress and some of them are just about done. So just to go through some of them, we're doing process development on the IVT. Oh, now I've broken it. How good. I actually have broken it. Hold on. Oh, there we go. Yeah, process development on the IVT, so improving the IVT process so we get less of, double-stranded RNA or improving the yield. And alongside that, we have the team is developing a double-stranded RNA ELISA assay to help us with that process development, so we know where we're at. And alongside that, the RNA purification methods, we're trying a range of those. On the LNP side of things, we're looking at freezing and freeze-drying for long-term storage, which I'll talk about in a moment, but looking at the next slide, it looks like it hasn't come up. PCR, so the facility is considering moving to PCR template production to get away from the problems that we have with plasmids, which Becca will talk about. And then in the future, we'll start looking at endotoxin testing of LNPs and rolling out sort of a preformed vesicle format where you can sort of have off-the-shelf LNP products that you can just add your RNA to when you're ready. So long-term storage of LNPs. So at the moment, any LNPs that you get from us are stored in the fridge, and we think they're sort of stable up to about 2 months, and Fiona has shown us that that's true. And hopefully she'll talk about that today. So long-term storage reduces mRNA waste. So when we make the lipid nanoparticles by microfluidics, you lose quite a lot of RNA. And obviously if your product expires, you also get rid of it then. So being able to store it long-term is a good goal for us. And you can achieve this by freezing or lyophilizing the LNPs, and that should also help us with our shipping conditions. So we ship things overnight or same day at the moment because it has to be kept cold, but if we've got lyophilized LNPs, then they can be shipped at room temperature. So just to show you some initial data from our freeze-drying experiments, we've made a bit of progress on this so far. So just looking at in vitro protein expression of GFP and HeLa cells, our lyophilized LNP is not performing too bad compared to a fresh control. And we do know that they work in vivo in a vaccine setting. So our main problem with freeze-drying LNPs is the encapsulation efficiency, so we're not looking too bad. And the antibody response is looking almost equivalent to the fresh control. And just to point out that there were a lot of people involved in doing this work from the University of Otago and the Mulligan and the Ferrier. Over to you.
So another thing that we've been focusing on lately is switching to a PCR template, or at least making sure that the facility is able to use both a linearized plasmid and a PCR template, because some of these techniques are more applicable to small scale and some to larger scale. But we were noticing that, like many people in the world, that we were having a lot of problems with plasmids that contain long stretches of A's, and these tails were concatenating and causing bigger and bigger plasmids that were getting harder and harder to untangle. And we know that's a problem worldwide, and some groups have moved to PCR to overcome this at small scale. So it has a few benefits, like you no longer need to involve bacterial cells, and so you remove endotoxin risk from your production. It's much faster, and we can start from a G block, so we can order a synthetic sequence of DNA, and that can arrive in 2 weeks, which is much faster than the process that we had to put new sequences into a plasmid. And your tail is much more homogeneous due to adding it on with a primer, which is what you can just see in this simple image here. So we just start with our synthetic DNA that we order in or could make, and not that we can now, but something we could think about in the future. And you amplify it with a primer that contains the T7 promoter as a tail that's needed to do our IVT reaction, and an ultramer which has the poly T tail which adds A's onto your product. So that allows a lot more flexibility. If we use the same UTRs for all our constructs, which we mostly do, you can use the same primers, and it makes the workflow much faster. And our awesome immunogenicity team did a comparison for us of RNA made from the plasmid template and the PCR template, and we just wanted to show that the responses are the same in vivo and that we didn't cause any major disruption. The product looked the same in all our QC, and this shows that there's no significant difference between the number of specific T cells, so T cells that had a specific response to our antigen, and also the same with the antibodies here. You can see that they are the same between conditions. So in a way, it's not a very exciting result to look at, but it was really exciting for us because it meant we could make this change. And so something else that the development team that I mentioned finds important is looking into new technologies that we could incorporate into our workflow. And so these are just some examples of people that we're working with. We're really excited to have a cloud spec machine that's there in the lab, and this is some of their team. They're from Marama Labs. They came out of Victoria University and have developed a machine that can read the concentration of RNA using absorbance in cloudy substances. It was developed for wine, but they've been applying it to LMPs, and so we're working with them, and it's really nice to have close collaborators and maybe an exciting new technology. Hurricane here is an example of a machine that will run IVT using continuous flow. So the idea is to have the template production, RNA production, all in one, producing a high quality product with a machine that could hopefully be easily scaled up to GMP. And that's with Harry Al-Wasiti from Monash in Melbourne. And we're hoping to get a pilot machine when that is available and start running some tests for them. And Precision Chroma is Sean Feast. He's located at the University of Canterbury, and he 3D prints chromatography columns, which are known to be really, really good for going from small scale to large scale due to the way they print the structure of your chromatography material. So that's someone we're thinking about collaborating with. So just to give an example that we're always on the lookout for new technologies as well and incorporating them in and giving them a try, and we're happy to talk about those or any that you've heard of that you think would fit in our workflow. Oh, I'm always going the wrong way. It's down, not up. So this slide is just to show that the way that we really work is still through having conversations, so main emphasis on this icon here. So if you want to work with us or you think using RNA might be helpful in your research, please get in touch. We are running the Fast Start projects, but also able to take on some external people into our workflow, and we just want to hear what you want to do and hopefully help you achieve your goals. And we're looking at different models for incorporating this in, so if we charge products at cost or how we can help to get you started. And once we've had those conversations, we send you products that we hope you'll have success with in the lab. It's been so far so good, and then emphasis on that coming back to us, and we like to turn things into a project and actually run multiple iterations to help you achieve your goal.
So just to give you an overview of what our standard products are at the moment, we can obviously do some of those things I talked about earlier with our new capabilities, but this is what you would get as standard unless you asked for something else. So the RNA, the mRNA is made by IVT, and generally we're incorporating a clean cap structure and a vector-encoded poly-A tail. As default, we're incorporating UTRs from the Pfizer-BioNTech mRNA vaccine, and then you can insert your gene of interest. And we can routinely do removal of double-stranded RNA if it's for a vaccine or going into mice, FPLC purification, and we'll look at the RNA integrity and purity analysis, and we can sort of do up to about 5 mg scale and down to 100 micrograms. For oligonucleotides, we can do a mixture of RNA and DNA, modified bases, the phosphorothioate or natural backbone, and kind of up to 30 nucleotides is kind of our limit at the moment. And so these are synthesized by solid phase oligonucleotide synthesis, and they're purified to greater than 95% purity, and we make sure that the salt is down by qNMR and things like that, and we can make sort of tens of milligram scale for those. And the lipid nanoparticles, as a standard, we're kind of using the Pfizer-BioNTech LNP recipe as a benchmark for everything, so you'll get that unless you ask for something else. And that generally has a size of around 100 nanometers and greater than 80% encapsulation efficiency, and we make sure we look at all the QC like encapsulation efficiency and size, and we can encapsulate up to about 1 milligram of RNA at the moment. And so we're not just working on platform projects, we can also take on external projects, as Becca mentioned, and this is just a couple of examples of the things that we're already working on or starting to engage with. So things like gene editing, skin delivery of RNA. We've been talking to Otago, for the avian influenza diagnostics, looking at RNA delivery to vines, flu vaccines for the elderly, understanding B-cell responses from RNA vaccines. We can also just encapsulate your RNA if you've got something fancy like circular RNA, and we're also looking at how to target dendritic cells for RNA delivery. So that brings us to the end, and there's lots of people to thank because there's been a lot of people involved in this so far and lots of businesses. So thank you very much, and we're happy to take questions.
Alex. You mentioned small molecule editors, so I'm wondering if you have the capability to